Okay, there we go. I'm going to go ahead and post this to our Facebook page and to our website once I get the recordings process. Usually takes about 48 hours or so for that. And then we'll post those up there for as long as we can. In addition to that, we have a Q&A at the end, which will be up as well. I do want to kind of go over kind of the legalese right here with this, is that we are not directly endorsing any of the products or claims made by any of the representatives. The recommendations that we do make about these products are based on current research findings, our best management practice findings and that type of thing, and are not influenced by the manufacturers whatsoever. And then finally, University of B Squad was not paid by the manufacturers for any of the webinar time. In other words, this is something that we put together on our own. The manufacturers are not paying for this, so it is completely for your information. Don't think of this as an infomercial, think of this as an informational session. That all being said, I do want to direct you guys to the next slide, which is upcoming webinars. So this isn't just a Veto Pharma. We're actually invited all the major manufacturers of miticides to come on and talk about their products. So next Thursday, we will have Beta Tech on, who makes HopGuard 1 and 2. Then on February 11th, we'll have Vita Europe, which is the producers of among other things, different thymol products, in addition to a new BT frame treatment that is out now in the United States. March 11th, we'll have natural or not apiary products, the Canadian group that has such products as Vitaway Quick Strips. And then if you want to register for any of these, the registrations are now open. And so you can go to the umdbsquad.com, look under upcoming classes and register there. I would recommend, if you're interested, to register early. We do have, I think, a 300-person cap on our license right now for Zoom. So once that fills up, we won't be able to admit any more people in. So if you're interested in any of these, please go ahead and sign up for them ahead of time. And then that way you'll be able to get into them and hopefully get some good information out of it. Another thing that I want to point out to you guys is, due to the number of people we have on the webinar, it's not going to be very practical to do the Q&A completely through the chat. So what I've done is ahead of time ask a lot of the beekeepers to send in questions. So we have about, I think about 15 questions that I distilled down from all the emails that came in. And so hopefully we'll answer a majority of your questions through that. If you have a question that's not answered, we will try to capture that in the chat after we do the initial Q&A. That being said, if we are not able to answer your question in the time allotted, feel free to email me, and I'm sure Freddie will have an email up as well for him. And we will add that to the list of questions to be answered if possible, but we'll do that at a later time. And so we really hope that we can answer all your questions. We've had some absolutely fantastic questions come in, so I'm looking forward to getting some answers for those. Finally, one thing I want to point you towards, and I'm sure Freddie will talk about this as well because he's involved in this effort, is the Honeybee Health Coalition. Most of the information you're going to hear tonight, hopefully you can find on the Honeybee Health Coalition's website in one of their products, the best management practices for hive health or the tools for Varroa management. They also have a Varroa management decision tool. It's an online tool for helping you decide what treatments are for your colony at a specific colony condition time. And so we'll definitely dive more into that later. Probably after the manufacturer series webinars, I'll do a webinar on choosing mite treatments, getting you guys ready for spring, that type of thing. I do want to point out, if you have ideas for webinars or subjects that you think would be really cool to discuss, feel free to email me at bsquad@umd.edu. And so I think that would be a great way to move into and develop future webinars. So without any further ado, I want to introduce our guest speaker. Let me just let one more person in here real quick, and then I'm going to come over to this. So Freddy is our speaker tonight. Freddy joined Veto Pharma a few years ago. He is currently an ESA certified master beekeeper and is the North American and Japanese manager for Veto Pharma. He also serves as the North Carolina State Beekeepers Association first vice president. He is a former full-time beekeeper with an extensive background in business who has over 3 decades of experience in honey production, commercial honey brokering, packing, and queen rearing. Freddie is a member of the North Carolina State Farm Bureau Beekeeper, Pollinator, and Honey Advisory Committee. He's also a county apiculture chair and represents the industry as a working group member of the Honeybee Health Coalition. His focus is in the business of beekeeping. So without further ado, Freddie, if you want to, I'm going to stop sharing my screen right now. You can take over and you can take it away.
Thanks a lot, Mark. I really appreciate it. So let me pull up a presentation and let's see if this works. All right. So are we up, Mark? Is this showing?
Not yet. Let me make sure your permissions are.
After doing like 250 of these. Thank you all for your patience.
Freddie, I went and upgraded your permissions to host, so that should give you full access.
Okay, so let me try this again. Share the screen.
And actually, if you wouldn't mind clicking on mine in the participants and making me co-host, then that'll allow me to do controls on my end.
Okay, so are you able to see this now?
Yep.
All right. Okay, are we good, Mark?
If you can make me co-host, then I can do the wrangling from there.
How to do that here.
Sorry, if you go to participants and on my name, you'll see more to the side of it and just click co-host.
Yeah. There you go.
Okay. Thank you so much.
All right.
And it's all yours, Freddie.
Well, thank you so much, Mark. I really appreciate it. And good evening, all, from the great state of blustery, cold winter North Carolina. It's good to see a lot of you faces out there. I certainly miss seeing you at all the different conferences, bopping around the U.S. at the different bee associations. And it's really good to see many of you again. Some of you I've seen every night for the last few weeks. So thank you definitely for allowing us to talk about beekeeping and certainly some of the products that are out there in the world of beekeeping. I also want to acknowledge tonight one of my colleagues, Mr. Phil Kraft, has joined us. And Phil, for most of you that do know him, but the few that don't, Phil is the North American technical consultant for Veto Pharma. He has been with the company for a number of years, and you'll see him at many of the conferences. He is a quite reputable speaker. He has been around for quite a few years, a former Kentucky State inspector, and certainly well known for his column, which appeared for quite a while in Bee Culture, called Ask Phil. So we appreciate Phil. I am the business side of Veto Pharma. As Mark mentioned, I represent North America and Japan for Veto Pharma. I manage these two regions for them, which means that not only do I deal with sales and business processes, but I am a liaison between corporate, which is located in Paris, France, a suburb of Paris, France, and deal with all different areas of beekeeping. So to start a little bit about Veto Pharma, we are a company that is based in France that has been around for about 30 years, 3 decades. And we are well known for our flagship product known as Apovar. Veto Pharma is located in a place about 3 hours, depending on how fast you drive, in Chaillac, France, with a very large facility for manufacturing Apovar.
Freddie, I'm sorry to interrupt you. Can you put your presentation into slideshow mode and it'll make the slides bigger?
Yeah, I did that here. So hold on. I'm trying, it's not seeming to work. All right. For some reason, it won't go into slideshow, Mark.
Okay, no problem.
Yeah, I will not, for some reason, go into slideshow tonight. All right. So to continue, so Veto Pharma is based in a suburb of Paris, France, called Palaiseau. That is where our corporate office is. And our manufacturing facility is located in a place called Chaillac, France. And this facility here is dedicated to creating the flagship product known as Apovar. We have about 80 people that work at Veto Pharma. And it's a very interesting process because we are classified as a pharmaceutical. Even though in the United States, Apovar is considered a pesticide, in Europe, Apovar is a medication, which requires a vet prescription in order to purchase. So we're very fortunate in the United States when it comes to beekeeping to be able to go down to our local brick and mortar or Amazon or our favorite online retailer and go ahead and purchase the products that we require without going through a veterinary process. At Veto Pharma, to give you an idea, we have a marketing team, we have a regulatory team, we have accounting, finance, a global sales team, a technical team, and a plethora of other areas such as quality control, warehousing, inventory, etc. Standard company. To give you a little idea of what it takes to be a pharmaceutical, and I'll just kind of walk you through a very quick process of a raw material delivery coming into the company. So the truck backs up to the incoming door, paperwork is signed, and product is admitted into the facility. At that very point, that pallet of product is brought into a special room, which is closed off and is put under negative pressure. A suited individual comes in, sprays off the pallet with compressed air, and this removes any dust, dirt, or debris before the raw material is brought into the facility. And this could be anything from a pallet of Varroa Easy Checks to raw materials, to other things that are coming into the facility. Once it is brushed off and it is cleaned off, it is documented, it is noted, and it is set aside. Samples of the raw material are taken and they are sent to quality assurance. At that point, the raw material or perhaps a completed product that is coming in has to meet specification. It sits into a quarantine until the process of quality control confirms that it meets the ordering and quality specifications. After that point, if everything jives, the product is brought into a warehouse and it can start the process of becoming or transiting through the warehouse depending on where it's going. Apovar is made by a batch. There's only 2 components to Apovar, but every single step that takes place in this facility is documented. So there is a chain of evidence. Everything is done by batch. Everything is done with documentation. And that's why it's very important when people look at their Apovar label, they realize that that batch number allows us to look at the quality and to associate a package that you have in hand with the process that we have gone through. It's a very labor-intensive process. So Apovar is made through an extrusion process where plastic and amitraz are blended and they are extruded through a special machine, many different heating points on that machine, and the famous strip that you know is produced. That strip exits the machine, it goes to a packaging area, and it is packaged. The label is placed on it, it is vacuum packed, and then it is labor inscribed or printed with the appropriate information, since it is classified as a medicine in Europe. The packaging that you see, and one of the questions that we'll address later on is, once I open a package of Apovar, how long will it last? And you have to realize that this packaging is specially formulated. It is designed to keep the amitraz in the Apovar strip from being activated. Once the product is produced, samples are taken, it's gone to quality assurance. The product may sit for 2 weeks, 3 weeks, 4 weeks, 5 weeks, 6 weeks until it meets the specifications of quality assurance, and then it will be released. Once it is released, then it can be shipped to the final destination. So that'll give you a little bit of insight into the facility, the pharmaceutical facility of Veto Pharma. Before we talk a little bit about Apovar and Apolifevar and oxalic acid, the products that we either manufacture or we distribute, we're going to start off a little bit with the Varroa Easy Check. The Varroa Easy Check, as you have seen, is a very popular device, and it allows beekeepers to get an idea of their mite thresholds. It's easily identifiable with the famous yellow cap. A lot of people enjoy it. It is considered a tool next to a hive tool in the bee yard. For several years, the Varroa Easy Check was exclusive to just alcohol wash. After a lot of internal investigation, trying to be ingenious like beekeepers normally are, and testing, we have found that we can use the components of the Varroa Easy Check that you have in your arsenal right now to accommodate sugar roll and CO2 injection. One of the things, and we'll talk about that in a second, is we've also given the Varroa Easy Check a little bit of a facelift. As you remember, it used to come in a little poly bag with bubbles on there, certainly fun for the kids to pop. That bag certainly came in handy in the truck, but it was a little bit difficult to stack in the store and to ship. So we now have in multiple languages a cardboard corrugated box, full 4-color, that allows retailers to better stack the product, to accommodate shipping a little bit better in the logistics supply chain. And it certainly works quite well when we are trying to aid retailers with a buying point. So I don't mean to be mundane, but we'll quickly go over the Varroa Easy Check alcohol wash. This is the process that we initiated at Veto Pharma for several years. And we believed at the time, and still believe it is the most accurate method to measure the number of mites per 100 bees. The way this works is we'll take a Varroa Easy Check, open it up, and remove the basket. We will fill it to about halfway or a little bit more. There's actually a line with rubbing alcohol or windshield washer solution. We will then take the basket and collect about 300 bees in there, which is about half a cup. And there's a couple ways of doing this. The first way is to take the basket and to run it at an angle down the frame. The bees will actually roll off into the basket. There are marks inside that basket for 200 and 300 bees respectively. And then you can submerge, once you reach the number of bees, estimated bees you desire, into the alcohol solution. Place the cover on, screw it on tight, and either shake or swirl, whatever your preference is. We want to do that thoroughly for about a minute. Yes, this procedure does euthanize the bees, but it also offers the best methodology for the bees to get washed. So Mark, may I interrupt and ask, are you able to see this alcohol wash screen?
Yes, yes, we can see it.
All right, thank you. After you have washed for about a minute, you'll hold the Varroa Easy Check up at an angle, and you'll take a look at the number of mites that are located on the bottom of the Varroa Easy Check. There are two trains of thought when it comes to using this methodology. Number one is we have the beekeeper who is very, very precise in counting all of the number of mites. And then we have the beekeeper that uses this as a visual aid and a tool to determine where they stand with their mite population levels. So anything under 3 mites, pretty good. 3 is kind of the threshold. Anything over 3, we're starting to get in the trouble area, and much more than 3, we have to address and utilize some form of treatment. Now, I can tell you from experience, Phil and I have done countless of these in backyard beekeepers as well as on the road with commercial beekeepers. And I personally prefer to use rubbing alcohol. A lot of people will consider using windshield washer solution, the blue stuff, preferably the winter mix. But what I have noticed is that eventually the winter mix becomes a little bit cloudy. It seems to dissolve more particulates off of the bees, and that rapid shaking motion seems to foam up pretty quickly. The rubbing alcohol through filtering methodology will allow me to get about 10 uses out of the same volume of rubbing alcohol. And here is an example where I'm just using a kitchen strainer and a paper towel. You could certainly use a coffee filter, it works just as well. But this allows us to reuse the liquid and to be a little bit more efficient in our bee yards. Let's move forward onto the sugar shake. So we felt that the Varroa Easy Check was a tool that just needed a little ingenuity for use. And we know that many people do not want to euthanize their honeybees. So where is that market? How do we deal with it? And how do we please the beekeeper, the need for that, and also allow them to maintain the lives of their honeybees? So in this method, by using the Varroa Easy Check as a sugar shake, we are flipping the basket upside down. So we'll take a Varroa Easy Check bin, and we'll put about 2 tablespoons of confectionery sugar in there. We will then use the base of the Varroa Easy Check, and we'll collect our bees either by rolling that container down a frame. And it's important that I reiterate that we're looking at brood frames that we're collecting these bees from. And we're looking at about 300 bees or half a cup. Another method is to take a frame or 2 from the center of the hive, the brood area, and gently shake the bees off into a container or a bucket and use a half cup measuring device, and then place your bees into the powdered syrup, or excuse me, powdered sugar mix. You'll go ahead and you'll take your Varroa Easy Check basket and you'll flip it upside down. So essentially we have something similar to a Parmesan shaker that you'd find at the Italian restaurant. Or for those of you more daring, a little vodka shaker that allows us to strain our goodies from the ice. The cap will actually screw on, and we can shake our Varroa Easy Check for about a minute. just like you would do with a mason jar or a plastic jar, standard screened sugar shake method. When we are done, there's a couple options. You can open up the Varroa Easy Check and use the cap to shake the remnants of the sugar and the Varroa mites into the cap. Or you can get another type of container. Recently, we used a white plastic plate out for great photography reasons. Sometimes, it's whatever you may have around. But we're shaking off the sugar and the dislodged Varroa mites into that. And then we're pouring a little bit of water onto the powdered sugar to dissolve the powdered sugar. This will allow us to see the number of Varroa mites. And we either have a choice of counting them or using it just as an indicator on whether or not where we stand with our thresholds. Finally, when we're complete with that process and we're satisfied, we can go ahead and we can add our live bees back to our colony that we removed them from, and also properly dispose of the mites as they will be swimming in a little bit of water. The third process is the carbon dioxide shake. And this is very similar to that of the sugar shake. Collecting the bees, putting them in the vessel, flipping the basket upside down. But we also are providing as an accessory, a CO2 injector. And we'll simply take the cap, just kind of put it on at an angle, not screw it on, and we'll inject some carbon dioxide. This does not kill the bees. All it does is anesthetize them. It puts them to sleep temporarily. You'll notice that the bees will become very active and then they will become very sedated. Screw the cap on, and we want to shake for about 10 to 15 seconds. You will notice the mites. You'll notice the mites will dislodge themselves from the bees during the shaking process. And we'll do the same thing as we did with the sugar shake. We'll flip it upside down. We'll shake it like a Parmesan shaker, and we'll be dislodging mites into the cap. The bees, of course, can be returned back to the hive. But the most important thing to realize about this method is that your mites will wake up and they are still alive. So we have to ensure that we don't return our mites back to our hives. We do not expect, just as a side note, in the United States, the CO2 to be extremely popular based on our culture and our methodology in beekeeping. We do know that sugar shake is extremely popular as well as alcohol wash, but our European counterparts and many other countries around the world are advocates of CO2. So we have offered this option. And the final thing for the Varroa Easy Check is some of you may be using some DFM products. By filling your unused Varroa Easy Check with some microbial product and flipping the basket upside down, it also makes for a great shaker to distribute product amongst the beehives. So we're going to jump from that to Apovar. So Apovar, all of you should know this, is the flagship product of Veto Pharma, and it was registered in the United States in 2012. It is the only authorized Varroa control product that contains amitraz as the active ingredient. The way this works is amitraz is a racocide and an insecticide that damages the nervous systems of the mites. It's a contact miticide where the Varroa mite will rub up, receive part of the molecule of amitraz, receive the amitraz molecule, and then eventually become paralyzed. The mite will drop and then suffer its own demise. the amount of amitraz in the strips is half a gram, or 3.33%, which is very, very low. And again, it's a contact miticide. And the temperature, it's not temperature dependent, meaning that we can use it in cold, warm, and hot temperatures. So unlike vapor-related miticides out there, this gives us a little bit more range depending on the time of year or the region that our bees may be in. Apovar, of course, has two ingredients. It has a polymer and it has amitraz. As simple as it can get. It sounds simple, but there's an awful lot of research that has gone into developing the migration of amitraz through that strip. The strip is placed in the brood chamber and it is inserted between frames in the hive. We normally like to see 1 or 2 frames difference. We don't want strips on the same bee frame space. We want them a couple frames apart. But most importantly, we want them in the brood chamber. One of the questions that we get all the time is, I've opened up my Apovar pack. How long do I have to use it? Can I delay it? Can I put it in the freezer? Can I put it in a Ziploc bag? And the answer is the packaging, like Apovar, is special. Once the vacuum sealed package is opened, we have activated the Apovar strips. So if we look at the timing of Apovar, so we have a 6 to 8 week treatment period using Apovar. If you're using it for a 6-week treatment and you open the package up and it's been open for 2 weeks, you're okay. So you've got to take into consideration to use it as quickly as you can. We get questions of, I've saved the Apovar, can I reuse the strips? And the answer is, please just properly dispose of the strips after you're done with your treatment period. And we are encouraging people to take advantage of Apovar and utilize 8 weeks for a full treatment period. When we look at Apovar, we have to sometimes move the strip in order to meet the movement of the cluster. And by doing that, and according to label, this gives us a little bit more time to add 2 more weeks to the application for a maximum of 8 weeks. So strip placement is very important. We're placing the strip in the brood chamber, in the brood area. And it is 1 strip for 5 frames of bees. So what does that mean? That means that for every 5 frames of bees in a brood chamber, I should have 1 strip. So if I have a 10 frame brood chamber, I should have 2 strips if I have 10 frames of bees. If I have a single 8 frame chamber, I should still have 2 strips, because it's 1 strip for every 5 frame of bees. But in this case, we have 5 plus an additional 3, which would allow us to put a second strip in. The treatment duration of 42 to 56 days, again, 6 to 8 weeks. And this again is a contact with the honeybees. So some of the things that we have seen in the field is lack of best practices. Where Phil and I have visited beekeepers and we have noticed that they have undertreated. They have placed the strip between the very last frame and the brood chamber box. Perhaps it was for convenience. Perhaps the bees were nasty that day. We don't know. But the strips were not placed in the right location. And we need to use enough strips. Some of the things that we certainly notice is undertreatment, and I'll talk about that here in a minute. But the key is to remember 1 strip for 5 frames of bees. So for those that are using Apovar in a 5-frame nuke, that's 1 strip for the 5-frame nuke. If we are going to a deep and a medium, which is utilizing a brood chamber, a 1.5, if we want to talk slang, we would use a total of 3 strips for that case. And if we had 2 deeps that had 20 frames of bees, we instinctively, according to label instruction, would use 4 strips. We encourage people not to remove them early. And a lot of times we have to actually move the strips. So after the strips have been inserted, it's good to go back and take a look at the health of your colony and to look where the strip and the cluster is. So after 3 to 5 weeks, sometimes we will notice that the cluster has moved where we have originally placed the strip. They're no longer present. So we remove the strip and of course use safety, wear some nitrile gloves, latex or disposable gloves, and remove the strip. Inspect it to see if it has propolis on there. If it has propolis, go ahead and use your hive tool, scrape some of the propolis off, and then move that strip over to where you see the cluster is. Repeat that process for every strip that you have in the hive. This will allow for more contact and future contact with the bees, since this is a contact miticide. For extra strips that you may not use immediately, we recommend keeping in a cool, dark, dry place, and to please use them as quickly as possible. And certainly keep them out of the sunlight. Sometimes we have a habit of placing them on the dash of the car, and it's very hot outside. Not recommended. One of the other things that we have to address when it comes to Apovar is that the label is king. As Veto Pharma representatives and as beekeepers who are advocates to other beekeepers, we can only specify what is written on the label. I know many times we are encouraged to go ahead and to bend the rules, to have our own personal opinion or experience, or to take a look what is posted out on social media or the bee forums. And we see a lot of information that is incorrect and is misguided. And as ambassadors to individuals in the beekeeping world, those that have been in for a while, who perhaps have not used the products that we're familiar with, or new beekeepers that are entering this industry, it is paramount that we share and we educate what is written exclusively on the label. From experience and being out in the field, if individuals had taken the time to read the label and install their Apovar or other products, treatment products correctly, taking into account for other products other than Apovar, temperature ranges, quantities, placement, we may have seen greater success rates amongst beekeepers. When we get into a situation where beekeepers say they may have had a treatment failure, the very first question we ask is, what are your monitoring documentation results? The Honeybee Health Coalition has been extremely instrumental in putting together a guide and recommending 3 to 4 monitoring times per year at minimum. We do the same. But if we just base ourselves and we do not monitor, we have no idea what the status of our mite levels are in our hive. Traditionally, we work off of a calendar. Beekeepers seem to always say, oh, when that blossoms or when that blossom falls, the honey flow is over. And we know in a few weeks we have to do treatment. Oh, it is the 2nd week of August, or the sun is in a certain position, or we see a wonderful twinkle in the stars, or the river's kind of turning a different way. And that indicates by calendar time, it is the appropriate time to treat. Unfortunately, that's no longer the case. Times have changed. Populations seem to be building up at different times, sometimes earlier. We've experienced this in North Carolina, where in the past, we could say, yes, we could treat in the 2nd week of August, but sometimes depending on the hive's population of mites, it now may be the 2nd week of June. And if we are not monitoring prior to treatment, post-treatment, during flow, we don't have an indication of where our colony stands. And one of the things that we do experience is the aftermath of the great honey flow. So beekeepers will extract their honey, and all of a sudden, they have forgotten about the mites. The concentration, and I'm to blame as well, I have done this for many years, goes on to the labor and task of pulling supers, extracting honey, and eventually we'll get back to the bees. And 2 weeks, 3 weeks is a very significant amount of time to where Varroa infestation levels can certainly increase while we are steadfast working at extracting our honey. And by the time we get our exhausted, weary bodies back out there in the hot temperature of the summer sun, sometimes we are too late. And it is good to have some documentation of what our mite levels were prior, during, and after treatment. One of the other things that we experience is reinfestation. We hear this all the time as beekeepers, and it is not specific to a Veto Pharma product. It is just specific to treatment in general, where beekeepers may follow a treatment regimen, and a few weeks after their treatment has completed and been removed, they noticed a reinfestation of mites. And sometimes that needs to be addressed, and secondary cleanup needs to be instituted. By having monitored results, we can actually take a look and see how effective the treatment was and when we need to treat again. The days of once a year treatment are long gone. And I can tell you, after being in this for 30 plus years, it was a pleasure in my youth to be able to neglect a hive of bees and not worry about anything other than the occasional wax moth or the occasional European foul brood outbreak you may get in spring. We did not have the pathogens, the scavengers, the things that we have nowadays, nor did we need the amount of skill when it came to honeybee husbandry. So it's changed in the last 3 decades. And we have to be aware that no matter how vigilant we are as beekeepers, there's outside influences that can affect the health and well-being of our hives. That can be reinfestation within the same apiary. We have witnessed in commercial operations where some pallets have forgotten to be treated and then have been a hot spot for mites, for reinfestation. You have neighbors that may or may not treat, depending on whether or not they're informed or if they have the time. And some beekeepers, unfortunately, just reinfest each other. It just happens. You know, many years ago, George Emery stated that there are those that keep bees and those that have bees. And the ones that have bees, no matter what you do, could be a danger to your hives. So it's important that you continue to monitor and that you continue to document. I'm going to jump quickly here to Apovar, but if memory serves me right, I may have a slide that shows the correct way that we insert an Apovar strip with the little triangle cutout. If I do not have that slide, when I cancel this screen sharing, I will give you a quick demo. So now we're going to talk a little bit about Apovar. Apovar is a thymol product that is manufactured in Italy and is distributed in over 35 countries. This is a very popular thymol-based product in Europe. Works extremely well. It is used as a primary as well as a secondary cleanup when it comes to Varroa mites. It has 4 components to it: thymol, eucalyptus oil, menthol, and camphor oil. And it works by contact as well as inhalation when it comes to the Varroa mite. Apovar has started to gain more and more popularity in the United States. And when do we use it and how do we use it? Apovar can be used in early spring, typically, depending on your region, well before the nectar flow, if the temperature conditions are within range. And yes, since this is a vapor application, there are temperature range, and those are indicated on the screen below as well as on the label. We can use it in summer and fall. Again, we have to be careful of region and temperature change. And we can use up to a maximum of 2 full treatments per year. We do not want to use this product when the external outside temperature is greater than 95 degrees Fahrenheit or lower than 64 degrees Fahrenheit. And the recommendation is to apply this early in the morning or late in the afternoon. It's extremely important when you use this product that you use gloves. It kind of feels and smells like Vicks Vapor Rub. And you know how that can get all over the place when we start touching a sweaty face or we start rubbing our hands. So it's very important that we take our safety and we have our PPE intact in our concern. When we open the package, which comes in a bright yellow sachet, we've noticed that many individuals like to open it up like a candy bar, grab the tab that is in the back and pull it down. The manufacturer suggests that we use a pair of scissors and just cut the top of the package off, which will allow us to slide 2 wafers out. You'll notice there are 2 wafers in there, and each one of these wafers will be broken up further into 4 wafers. So each package contains the 2 wafers. European side, they call them tablets. And we'll divide that tablet or wafer into 4 parts. We'll open the hive, and on the very top bars of the uppermost box, we will place one of these quarters of 1 tablet in the corner underneath the undercover or migratory cover. Again, we're paying attention to temperature, and we want to make sure that these tablets are placed in the corners and not directly over the brood chamber. We'll go ahead, we'll repeat this in 7 to 10 days. And then for the 3rd treatment, we'll leave it in up to 12 days. What we have seen is sometimes the bees initially will propolize the wafers, or they will try to remove them out of the colony. Sometimes they're successful, sometimes they're not. But they eventually get used to the vapor. Initially, you may notice that there may be some bearding on the bottom board, but again, that will go away once they get a little bit used to it. So the bottom line is the way this is packaged, in order to do a proper treatment, you'll need 1.5 sachets to ensure a total of a full treatment of 3 individual applications. So 3 bags will satisfy 2 colonies. And finally, we'll go to Apobioxal. Apobioxal is oxalic acid, but with us at Veto Pharma, this is a pharmaceutical grade oxalic acid. It is 97% OA and 3% inert. It is the only registered product for use in beehives in the United States. And there are only, at the moment, 3 approved methods for using this. Number 1 is sublimation, which sometimes people incorrectly call fumigation or vaporization. We also have drench, which can be called dribbling or trickle, depending on your vernacular, which region you come from. And finally, we have a spray methodology, which is used for swarms and packages. Apobioxal, based on the current label, utilizes only these 3 methods. Anything else that we see on YouTube or on social media violates the use of this product. It's imperative when we work with oxalic acid that we take our personal protection and safety at the highest level. There's nothing worse than getting oxalic acid crystals on wet, sweaty hands. It will burn. It hurts. I've done it. I've been there. So please, whenever you're mixing up a solution of sugar syrup with oxalic acid, please take the appropriate measures. Wear safety glasses to avoid splashing into the eyes, and certainly wear proper clothing. Do it in an area that is away from children, away from pets, and make sure that you label it properly, and don't store it in the kitchen fridge or the kitchen freezer without the proper label, and ensure that it is not easy to get a hold of. Shelf life in the dry form is 5 years from the manufacturing date, and it currently comes in 3 sizes: 35 gram, 175 gram, and 350 gram. I know we're not typically used to using grams, but 35 grams will mix into 1 liter of 1-to-1 solution for a dribble. And with that solution, we will take a total of 50 milliliters, and we will dribble a maximum of 5 milliliters per bee space with a maximum of 50 milliliters per hive. When we use it for sublimation, it is 1 gram per brood chamber. And the spray is simply mixing a 35 gram packet into a 1-to-1 sugar solution or 50% sucrose solution. Spraying according to the label. A 2-pound package, for example, is around 21 grams, if I remember correctly, and then letting the package sit for a specified amount of time. Most importantly, when it comes to oxalic acid, since it is a product that we will most likely mix, we have to be careful, and our safety is paramount. So that is the presentation I have. I will quickly show you, if I have an Apovar strip here, I do on my desk, the proper way to insert an Apovar strip, and then we can transition over to the questions. So can everybody see this?
It's good.
All right. So if you notice, this is first of all an inert Apovar strip. So I'm doing my due diligence to tell you this has no amitraz in it. This is a demonstration strip, and this is the actual plastic that we use. The strip normally comes with another strip attached, and we tear them apart to get our individual strip. If you'll notice on the top, there is a tiny little hole here. We can use a toothpick or a nail, and we can suspend a Apovar strip in the brood chamber, the center of the brood chamber, and between the frames. The other method that we can use is using this tiny little triangle. A lot of times when we have gloves on or we watch other beekeepers, we just will simply put the the Apovar strip in with the tab at about 90 degrees. And what this does is this allows the strip to push up based on the force of that tab. That's incorrect. What we want to do is we want to take the bottom of the triangle and we want to make a horseshoe out of it. And once you've done this a few times, it's very easy. This horseshoe now gets slid between the frame space of your Hoffman frames, and this is what holds your Apovar strip in place. So we see a lot of misuse where the strip is inserted like this, but the proper way, again, is to flip it like a horseshoe or a letter U and insert it down into the frames. And just as a reminder, when it comes to Apolite Var, this is the sachet. We do not want to tear it open like a candy bar. We will crush and destroy the wafers. We want to use a knife or preferably a pair of scissors, cut the top off, and then slide our 2 tablets or wafers out and then apply them to our colony. So Mark, I think that is about it for this quick product presentation. And if you'd like, we can certainly transfer over to the questions.
Yeah, that'd be perfect. So we've gotten some questions coming in the chat, but what we're going to do, guys, is we're going to hold off on those right now. And I'm going to share my screen, and we have some of the questions that came in ahead of time. And I think this will actually cover some of the questions that people had. So let me go ahead and share this. Okay. Make it full screen here. Is that loud and clear there, Freddie?
Yeah, that looks good.
Excellent, excellent. So we'll go ahead and let you, if you want to read through them and kind of give your answer to them. I think you've answered quite a few of these, but we can kind of refresh on them if you want.
Sure. Can drawn frames that have, in past season, been exposed to Apovar be used for honey frames and the honey extracted be consumed by humans? Well, that's 2 questions in one. The simple answer is yes. If Apovar has been used in compliance with the label and it's not been left in over the entire winter, this should not be a problem. We see that there is quick degradation of amitraz, including the amitraz metabolites, and they'll be degraded over to the following spring. However, you have to take into account that you want to exchange brood combs about every 3 years. So there is wax management and wax hygiene. And this also takes into account that it was just Apovar that you have used, and you have not used any other amitraz-based product. You have to remember, when it comes to the combs, there is no restriction that is listed on the label. And as we mentioned before, label is the law. Now, as a beekeeper, I personally have a little bit different viewpoint. As a beekeeper, I prefer to mark my frames that have come into contact with any sort of registered treatment and ensure that that frame, which was originally used for a honey super, now becomes a brood frame for the rest of its life until it is rendered. That is my own personal methodology, and that is what I prefer to use. Now, also, honey that is come into contact with Apovar strips, we should take that and we should feed that back to the bees, presuming it is our honey from our hives, and just leave it for the bees. So the next question is, what are the best times of year to treat with Apovar and oxalic acid? Very interesting question, because there's a couple trains of thought here. If we look at our commercial beekeepers, many of them treat with Apovar in the spring before the honey flow. And the reason for this is that they want to start the year off with the cleanest bees, the healthiest bees, and the least amount of mite population. On the hobbyist side, we seem to tend to see more individuals treat in the fall time. It's just perhaps something of habit. But again, calendar base versus the amount of threshold in a monitor is extremely important, and that dictates when we should use it. Now, oxalic acid, when we look at the label, the current label, it says that we treat when there is little to no brood present. So what does that mean? That means that the best time for us to utilize oxalic acid is in the winter months. So November, December, when your colony has the least amount of brood. Of course, we hear people that sublimate when there is high levels of brood, but this currently violates the label. Some of the things that we see people do in other countries is they will cage the queen in the summertime when they have high populations of honeybees for about 25 days. This allows brood to emerge, especially if there's any drone brood, and then they will be able to treat their colony. This allows them, through caging of the queen and restricting laying, an ability to reduce the amount of brood. So oxalic acid when there is little to no brood. Third question is, what is the correct dosing of Apovar for only 10 frame medium configuration? For 8 frame boxes, both deep and medium. So this is an interesting question, because are we talking 110 frame medium that is serving as an overwintered brood chamber in Florida? Are we talking a 2 frame medium that is overwintering in Kentucky as 2 brood chambers? Or are we talking 3 10 frame mediums that are sitting in the great state of New Hampshire, sitting as 3 brood chambers? It's a great question. There's lots of confusion. The correct answer is, for 1 to 2 individual medium boxes, it will be 2 strips of Apovar. Depending on colony strength, you could add a third, but reading by the label, it should be 2. We base that on the fact that 2 medium brood chambers are somewhat equivalent in surface area to a Dadant style hive, which is used